Hey, hello everyone, welcome back to my video channel. And as you guys already know, I'm going to solve the KSET Life Science question paper from 2013 to 222. Okay, and for this video the topic is methods in biology. So what I have collected all the questions that belong to this topic and I have made a list here. So I'm going to solve these questions and this video is part 2. Okay, if you didn't watch the part 1 video, you can go back and watch that video. Okay, thank you. So now let's start with the first one. Okay, here the first question is molecular, 13th question, continuation of that. The molecular karyotype means, which is as in the year 2015. Option A, pulse field gel electroporosis. Option B, polyacromidal gel electroporosis. Option C, agarose gel electroporosis. Option D, 2D electroporosis. Okay. So before that, let us know what is this molecular karyotype. Molecular karyotype is nothing but it is a CGH array, means it is a comparative genomic hybridization array. Okay, it is a new method in prenatal genetic testing, which can detect genetic malformation. Okay, malformation means if there is any abnormalities, okay, disorders in the fetus that wouldn't otherwise be detected before the childbirth. Okay, and this molecular karyotype compares the patient's genome against a reference genome and identifies the difference between the two genomes and hence locates the region of the genomic imbalance, means that if the patients have any malformations, any disorders in the patients. Okay, that's about the molecular karyotype. So they are asking molecular karyotype means. Option A, pulse field gel electroporosis. Let us know what is this pulse field gel electroporosis. Here this pulse field gel electroporosis is used to identify the DNA anomalies, DNA abnormalities. Okay, and it is a kind of technique used for the separation of larger DNA molecules by applying to a gel matrix on the electric field. Okay, on the electric field that periodically changes the directions. As I mentioned here, this technique is used about more than 10 MB DNA. Okay, and this is more effective than regular gels, regular techniques at achieving differential separation of the small and larger DNA molecules. This is because due to the constant changes in the direction of the electric field in the gel. Okay, that's why this pulse field gel electroporosis is more accurate one than the rest of the techniques. And here in this technique, the largest pieces of the DNA are subjected to pulse field gel electroporosis by applying electric current. Okay, by applying electric current, and the second is altering its direction at regular intervals. If you have to compare with the other electroporosis, you have to take the agarose gel electroporosis. This agarose gel electroporosis is done to separate the smaller fragments where the current is applied in a single direction, but in case it is in the multiple directions, in many directions. Okay, this is and using the larger pieces of the DNA, and this is using the smaller one. Okay, and if you have to say some few ideas on the second option, that is polyacromidal gel electroporosis, that is PAG. Okay, PAG. This is a technique widely used in biochemistry and in organic chemistry and in biotechnology to separate the biological macromolecules, like usually you can say proteins and nucleic acid. And this is based on, according to their electrophoretic mobility, means it is purely based on their size, mass. Okay, this one, remember, this is purely based on the size, mass. Okay, and for this agarose gel electrophoresis, like the smaller fragments migrate faster than the larger ones. Based on that, based on this principle, this agarose gel electrophoresis technique works. Okay, and the next one is 2D electrophoresis. This 2D electrophoresis, this is also analyzing the proteins. Okay, I mean, this technique actually, this 2D electrophoresis allows a separation of 100 to thousands of proteins in one gel. Okay, and this is going to separate the proteins. I mean, proteins are separated as per the isoelectric point and then the protein mass. Okay, so by this analysis, we by this a small information, we get to know what is molecular karyotype. Okay, and the little bit idea on all of these, right? So by this we get to know that if this is not particularly for anything, so this will be the wrong answer, because it is going to use for DNA, RNA, proteins, everything. Okay, and this is especially for the proteins again, and this one is also for protein. Here I said what is molecular karyotype. Okay, molecular karyotype is that is going to detect the genetic malformations, I mean disorders. So to identify those DNA anomalies is the technique that is used is pulse field gel electrophoresis. That's why option A is the correct answer. Okay, now let us move on to the next one, that is the probes for DNA fingerprinting are. So they are asking what kind of probes are used in DNA fingerprinting. Okay, which is asked in the year 2015, which is asked in the year 2015. And here, so in the lower classes itself, you have heard about this one, that is DNA fingerprinting, right? Why they are using this? They are it is mainly used in the forensic analysis. I mean, in to find out the criminals, right? So if you know the procedure, procedure of the DNA fingerprinting, you will get to know the answer for this. Okay, so let us know what is the. I mean, I will explain in very short. Okay, DNA fingerprint. This is also called as DNA profiling. Okay, and this one was discovered by Alec Jeffrey, and this is used to distinguish between the individuals of the same species using only the samples of their DNA. Okay, and we know that there is called as VNTR. Okay, VNTR. What is this VNTR? VNTR is nothing but a variable number of tandem repeats. Okay, this variable number of tandem repeats polymorphism is the basis of DNA fingerprinting, which are short nucleotide repeats. Okay, because these VNTRs, the location and repetitions of these VNTR is so unique that no two individuals are alike. Okay, and here the probes are used to analyze the, I mean, to test, to do the DNA fingerprinting. There is a special DNA probes are used. Those are single stranded labeled DNA. Okay, these probes are used to search for its complementary sequences in a sample genome. I mean, here DNA probes with the known sequence of the probes, I mean DNA probes, they have a known sequence of probes, right? Those are complementary to these on the VNTRs. Okay, and these are going to use bind to these VNTRs. So, and later they are going to make them radio labeled, and then they are doing the observations. Okay, so if there is any difference, they are going to find out the going to distinguish between the individuals by the differences. Okay, that is about the DNA fingerprinting. So while I am explaining, I said the probes, here they are asking about the probes. That's why they are using the known single standard labeled DNA. So that's why option C is the correct answer. Okay, now let us move on to the next question. Who got the Nobel Award in Medicine for devising the MRI technique? It is asked in the year 2015. Option A, H. Temin and D. Baltimore. Option B, R. Franklin and F. Crick. Option C, P. Laturber and P. Mansfield. Option D, G. Binning and E. Sutherland. Okay, so these are the scientist names. So if you know who invented this MRI technique, so you just know the scientist names, means you are going to get the marks for this. So and here the the persons who awarded Nobel Award in Medicine for devising this MRI technique is that the person is, that is option C is the correct. These two scientists are invented this MRI technique, and they awarded the Nobel Award in the year 2003. Okay, in the year 2003. And this MRI, what is this MRI? MRI means magnetic resonance imaging. Okay, here magnetic field gradients and radio waves creates the images of the organs of the body. Okay, so that's about the MRI. Now let us move on to the next one. The next question is, when a distribution is positively skewed, okay, option A, the mean is higher than the mode. Option B, the mean is lower than the mode. Option C, the mean is equal to mode. The mean is equal to, option D, the mean is equal to median, which is asked in the year 2015. Here, this comes under the statistics, okay. If you learn that in it, you can be able to answer this question. And in this case, they are asking positively skewed, okay. Before that, let us, I will just give a small idea on the what is this skewness, okay. What is this skewness here? Let us know the first meaning for this. So here I have drawn the picture here. I guess this is all visible, okay. So before explaining the skewness, let us know if there is no skew. So this is the diagram here. If there is no skew, that means that there is a symmetrical distribution, okay. And if there is a skewness, okay, if there is a skewness, here you can see if there is a skewness, it is asymmetric, means you can observe in the picture here, this is not asymmetric, okay. The distribution is shifted to right side, and distribution is shifted to left side, and distribution is shifted to right side, okay. This has been occurring like this, okay. If there is no skewness, that is systematic distribution. If there is a skewness, that means it is asymmetric distribution. So here there are two types of skewing, okay. That is negative skew, and then is the positive skew, okay. And if there is no skew, in that case, mean is equals to median, median equals to mode, okay. That means it is 0. Here I hope you guys know what is this mean, median and mode. Here mean means it is the sum divided by the count, and mode is the most repeated number, and median is middle number, right. Mean it actually gives the average. And here this skewness actually gives the data on the shape of the data, okay. It gives the shape of the data. And as I said here, there are two types in this. In case of negative skew, if you have to explain the negative skew, that is, it is also called as left skew. So here what happens, the distribution will be on the one side, okay, which is on the left side, okay. Here mean will be the lowest and mode will be the maximum. Here mean is less than median, okay. And in case of positively skew, here it is also called as right skew. Here in this case what happens, mean will be greater than median, median will be greater than mode, okay. Here they are asking, the question they are asking is about positively skewed. So positively skewed, as I explained just now, here as I said, if you look at the options here, the options should be, what we have to get here is mean should always be greater than median and the mode. So here the correct answer will be option A, that is mean is higher than the mode here, okay. This is the correct option, that is option A, okay. Now let us move on to the next question. The next question is on the standard deviation. So here the standard deviation is a measure of dash of a frequency distribution, which is asked in the year 2015. So it is very simple and direct question. If you know the definition and some points on the standard deviation, you can solve this question. So yeah, obviously the standard deviation is a measure of variability, okay. It is a very simple question, okay. The standard deviation is a measure of variability, okay. Now let us move on to the next question. Here the next question is like this, which is asked in the year 2015. Clones are identified by hybridizing them with, okay. So the correct answer for this is option A. The clones are identified by hybridizing them with the probes, not the vector. Vector is you may get not the with vector, not with antibodies and virus, okay. Clones are identified by hybridizing with the probes, okay. It is very basic question, okay. Now let us move on to the next question. Radiations can be detected through, which is asked in the year 2015. Option A, ELISA reader. Option B, spectrophotometer. Option C, Geiger-Müller counter. Option D, Mardi-Toff. So before that, let us know what are these ELISA reader. So what is this ELISA reader? So yesterday I have explained the ELISA, right? So here this ELISA reader, it is going to measure the antibody test, protein test, which is used for on the protein test, enzyme assay, and it also finding out whether the person is infected with HIV or not, and also used for quantification of the nucleic acid. Okay, and this ELISA reader is works on the principle of specific antibodies bind to the target antigen and detect the presence and quantity of the antibody binding. It works on the based on that. So there is they are asking radiation. So but in this ELISA reader, the main principle, if you have to say, the main principle is it is going to work on the principle of the specific antibodies bind to the target antigen. Okay, so that's why this is not the correct answer. And the next one is spectrophotometer. So this spectrophotometer, I guess I have explained in the previous video. I will repeat once again here. This spectrophotometer is a method to measure how much a chemical substance absorbs a light by measuring the intensity of the light as this beam of light passes through the sample solution. I mean, the basic principle of this spectrophotometer is that each compound absorbs or transmits light over a certain range of wavelength. Okay, so this is again not about the radiation. So this is also not a correct answer. And then now the third one, Geiger-Müller counter. This is also an electric instrument used for detecting and measuring the ionizing radiation. Okay, used to detect and measuring the ionizing radiations like alpha radiation, beta radiation, and gamma radiation. Okay, this is especially used for the radiation detection. Okay, so by this knowledge we get to know that option C is the correct answer. But let us know what is the meaning of this MALDI-TOF. Here this MALDI-TOF is used for viral identification and also for the mutation and in genotyping and to find out the antiviral resistance. And this actually uses the laser. Okay, so this is about this. So option C is the correct answer. Now let us move on to the next question. MRI is made possible by which of the following, which is asked in the year 2016? Option A, external magnet of high strength. Option B, gradient magnetic field. Option C, Fourier transformation of the NMR signal. Option D, improved radio frequency detectors. As I explained this MRI, so do you know what is the MRI long form, full form here? This is magnetic resonance imaging, right? So actually this MRI uses the magnetic field. Okay, this as I explained this, okay, while solving this question, that is while solving the 15th question for who got the Nobel Award in medicine for devising the MRI technique. So this uses the gradient magnetic field. Okay, that is the correct answer. Now let us move on to the next one, 21st one. A device used for measuring arterial pressure, which is asked in the year 2016. Option A, electrocardiogram. Option B, defibrillator. Option C, otoscope. Option D, sphygmomanometer. Okay, so to solve this, let us know what are these. So electrocardiogram, this actually measures the heart rate. Okay, so and then this defibrillator, actually this one, if you have to say this defibrillator, actually you guys are very familiar about this, because if you are more interested towards the movies, in movies where the hero or hero's mother or father are very serious and they have cardiac arrest, that time they will use this one, defibrillator. ನೋಡಿರ್ತೀರಾ ಇನ್ ಟಿ ವಿ ಶೋಸಲ್ಲಿ ಅಥವಾ ಮೀನ್ ಮೂವೀಸಲ್ಲಿ ಇದನ್ನು ಯೂಸ್ ಮಾಡ್ತಾರೆ ಎಗೇನೆಸ್ಟ್ ದ ಹಾರ್ಟ್ ಹತ್ರ ಪ್ರೆಸ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. Okay, so that they will respond to that. I mean this device that gives high energy electric shock to the heart of someone who is in the cardiac arrest. Okay, so that's why these two answers are not the correct answers. Then option C is also not the correct answer. Option D is the correct answer. Okay, that is sphygmomanometer. Okay, then we will move on to the next one. Okay, which is the least invasive method of the following used to detect genetic disorders before birth? So here option A, chronic villi sampling. Option B, amniocentesis. Option C, ultrasound imaging. Option D, fetoscopy. It is asked in the year 2016. So let us know what is this. Now, if you have to say fetoscopy, let's talk about fetoscopy. Feto means, we know that feto means child. Okay, this is used for the pregnant woman to find out about the child. Okay, to examine the unborn baby. Okay, what they will do in this fetoscopy, it is a surgical access to the fetus, the amniotic cavity, the umbilical cord, and the fetal side of the placenta. Okay, to there a small incision is made in the abdomen, and endoscope is inserted through the abdominal wall, and then abdominal onto the uterus into the amniotic cavity. From there, they are going to get the fluid and they are going to make the testing. That is about the fetoscopy. Okay, so you got to know about the fetoscopy, right? Now, option C, ultrasound imaging. Ultrasound imaging, I guess I explained in the previous video. Please do watch that, so that will make easy for you guys. So this ultrasound imaging actually it uses the sound waves. Okay, it uses the sound waves to find out any kind of abnormalities of the unborn child in the pregnant woman. Okay, this is also called as sonography. Okay, so in this case, ultrasound imaging, so there is no insertion is made. There is no need of insertion of the needles into the pregnant woman body. Okay, here in this case it is made somehow little. Okay, amniocentesis, again this is also a prenatal diagnosis of the chromosomal abnormalities and also fetus infection, and also by this amniocentesis technique there is a possible to find the sex determination also, means to find out whether the child is a boy or a girl. That also you can find out. Means in this technique what they will do, they uses the amniotic fluid is taken from the uterus, which is surrounded around the body. Okay, so in this case also a small invasive insertion is also made to the pregnant woman. Okay, the option A, chronic villi sampling. Here chronic villi sampling is also called as CVS. In this case what happens, the sample of chronic villi is taken from the placenta, where it is attached to the wall of the uterus, and by using an instrument called as catheter, it's like a needle is inserted into the lower abdomen to retrieve, to take the cell samples. Okay, here also invention is made. So by this we got to know this. The question, if you understand the question here, they are asking which is the least invasive method. Andre ಯಾವುದರಲ್ಲಿ ಇನ್ಸರ್ಷನ್ ಮಾಡಿಲ್ಲ. ಓಕೆ, ಲೆಸ್ ಆಗಿ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ ಅಂತ ಹೇಳೋದಾದರೆ ಈ ಮೂರನ್ನು ನೋಡಿದರೆ, ಈ ಮೂರು A, B and D, this insertion is made, okay, to find out the genetic disorders. But in case of ultrasound imaging, there is no any kind of needle instrument used. Okay, only by using the sound waves they are going to find out the genetic disorders. That's why option C is the correct answer. Okay, now let us move on to the next question. 23rd, identify the technique that allows thousands of genes in different types of cells to be compared simultaneously. It's asked in the year 2016. Option A, microarray. Option B, in situ hybridization. Option C, RFLP. Option D, restriction digestion. So before that, let us know what are the options. Okay, here question that has asked here is about the technique that allows the thousands of genes in different types of cells to be compared simultaneously. Okay, which is that technique they are asking. So option A, they are saying option D is restriction digestion they are saying. So I guess I have explained this, I have given idea for this in the previous video. Please do watch that video, but I will repeat once again here. See, it is very simple process. This is only it is the process of cutting the DNA molecules. Okay, it is the process of cutting the DNA molecules into a smaller pieces with special enzymes called as restriction endonucleases. Okay, so there is no comparison comes here. Okay, so that's why option D is not the correct answer. Okay, and the next one is if you have to go for the B option, that is in situ hybridization. So what is this in situ hybridization here? In situ hybridization, it is a procedure that allows us to locate the position of the specific DNA sequences on the chromosomes. Okay, that is the in situ aberration. Means this is the technique that relies on the exposing the chromosomes to a small DNA sequences that is called as probes that has a fluorescent molecule attached to it. So by that using that probe, they are going to locate the position of the specific DNA sequences on the chromosome. Okay, so that's why this is also not correct answer. Then the next one is RFLP. What is this RFLP? Form of RFLP is restriction fragment length polymorphism. Okay, this is a genetic analysis that allows the individual to be identify the pattern of restriction. To identify based on the unique pattern of restriction enzymes. Huh? This is the Forsenic method to analyze the DNA. Here, we know that the DNA of each organism has a specific sequences called as restriction fragments that can be cleaved by the restriction endonuclease enzymes to produce the fragments of different length. Okay, so this is about that. Okay, so this is not the one it is not comes to find out to compare simultaneously. That's why this procedure is also not a correct answer. So option A is the correct answer, that is microarray. So what is this microarray? I guess I have explained this microarray in previous video, that is don't mind like I'm again saying I explained the previous video. Definitely if you watch that video, because there I have because these questions, as I said before, the questions are going to repeat. Okay, so and the options, in that options there are so many techniques. I can't explain again and again because I've explained in the another video already. So while solving those questions, so the that's why I said if you learn like a few of the techniques, you can solve so many questions. That's why I will highly suggest you and recommend you to study the 13th chapter. Don't neglect that chapter, that is methods in biology. Study that because these are direct questions. If you just get to know the techniques, you can answer so many questions. Okay. So here microarray method it is a developing technology that is used to study the expression of the many genes at once. Okay, it is a tool used to determine whether the DNA from a particular individual contains mutations in gene or not. Okay, this is the one where the simultaneous comparison will be possible. So the correct answer is option A, that is microarray. Okay, now let us move on to the next question here. The next one is the functional genomics. The functional genomics can be best studied with, it is asked in the year 2016. Option A, PCR. Option B, DNA microchips. Option C, dot blot. Option D, ELISA plate reader. So now let us solve this. So here the option D, what they are saying, option D here is, it is ELISA plate reader. I guess I explained this ELISA plate reader, what they are using by ELISA plate reader. It uses the antibody and antigen, right? Okay, so antigen antibody use ಮಾಡಿಕೊಂಡು find out ಮಾಡ್ತೀವಿ. So that's why this is not the correct answer. They are saying about the functional genomics. ಯಾವ ಟೆಕ್ನಿಕ್ನಿಂದ ಫಂಕ್ಷನಲ್ ಜಿನೋಮ್ಸ್ನ ಚೆನ್ನಾಗಿ ಕಲಿಬೋದು ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೊ ಇಟ್ ಈಸ್ ನಾಟ್ ಅಬೌಟ್ ಇದರಿಂದ ನಾವು ಫಂಕ್ಷನಲ್ ಜಿನೋಮಿಕ್ಸ್ನ ಸ್ಟಡಿ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಓಕೆ, ನಾವು if you go for the option A here, PCR, it is also it is also called as, the full form for this is polymerase chain reaction. Okay, this is actually used to multiply the DNA copies. Okay, if you have to say in more particular, polymerase chain reaction is it is a chemical reaction that used to amplify the pieces of the DNA. This polymerase chain reaction allows a single or few copies of DNA to be replicated into millions of billions of copies. Okay, by this also we can study the functional genomics here. So now the next one is that is DNA microchips. Okay, DNA microchips, these are nothing but microscope microarrays are nothing but these are microscope slides that are printed with thousands of tiny spots in a defined position, with each spots containing a known DNA sequence or the gene. This is the tool used to determine whether the DNA from a particular individual contains a mutation in genes or not. Okay, for that reason they are using this DNA microchips. Okay, so next one is the dot blot. This is used to detect the protein. Okay, this test is used for the presence to find out the presence of protein of interest in a sample or not. Okay, and this dot blot is the simplified version of the Western blot method. In case of dot blot, there is no electrophoresis step. Okay, there is no electrophoretic step. Okay, it is a simplified form of the Western blot. Okay, so as they are asking about the functional genomics, so I guess do you know what is this functional genomics means? Functional genomics means it is the study of or you can say studying the expression and functions of the genome. I mean, like whole structure, functions and regulations of the gene. So everything is studied in the functional genomics. Okay, so that is the definition of the functional genomics. So ಅದನ್ನು ನಾವು ಯಾವ ಟೆಸ್ಟಿಂದ ಚೆನ್ನಾಗಿ ಕಲಿಬೋದು ಅಂತ ಕೇಳ್ತಾ ಇದ್ದಾರೆ. ಫಾರ್ ದಿಸ್ ಆಪ್ಷನ್ ಬಿ ಇಸ್ ದ ಕರೆಕ್ಟ್ ಆನ್ಸರ್, ದಟ್ ಈಸ್ DNA microchips. Okay, here you may now get confused with the DNA microchips and microarray, because this DNA microchips is also called as DNA microarray. Okay, so but there is a difference between microarray and DNA microchips or DNA microarray. Okay, so here this microarray, okay, it is a laboratory method tool used to detect the expression of the thousands of genes at the same time. Okay, but these DNA microchips or the DNA microarray are the microscopic slides that are printed with thousands of tiny spots in a defined position, with each spot containing a known DNA sequence or the genes. I mean, this is used to determine whether the DNA from a particular individual contains a mutation in genes or not. Okay, so that's why this is the correct answer. Means here you can see by this you can find out the you can study about the functional genomics using this technique. Okay, now let us move on to the next question. In which of the following technique there is no electrophoretic step, which is asked in the 2017. So while I am explaining the 24th one, I guess I have given the answer for this also. So it is very easy now to answer. Option A, dot blot. Option B, Western blot. Option C, Northern blot. Option D, Southern blot. So here option A is the correct answer. So we know that what is a Western blot. Western blot is to analyze the protein, Southern blot is to analyze the DNA, and Northern blot is for the analyzing the RNA. So here the dot blot is the correct answer. So dot blot is also it is to identify, to detect the protein only, but there is no electrophoretic step in the dot blot. Okay, so that's why this is the correct answer. Now let us move on to the next one. In Poisson distribution, so dash, which is asked in the year 2017. Option A, mean is greater than variances. Option B, mean is less than variances. Option C, mean is equal to variances. Option D, mean is greater than standard deviation. So this is a question from the statistics. Okay, here let us before solving this, let us know what is this Poisson distribution. The Poisson distribution is nothing but it is a probability distribution that is used to show how many times an event is like to occur over a specific period of time. Okay, in case of this Poisson distribution, here the mean is always equal to the variances. Okay, the mean is equal to the variances. Okay, the correct answer for this is C, that is mean is always equal to the variances. Okay, so that's about this video, and this is the part 2 video, and I have still some more questions. Okay, I will solve these questions in the next video as a part 3 video, and I'm hoping that this video is helpful for you all. And if you learn something new from this video, please do subscribe and share. And thank you everyone. Have a nice day.